Today we'll talk about genetic basis of antibody structure, and objectives of today's lecture are structure and gene expression of antibody and genetic events in the synthesis of immunoglobulin chains, class and isotype switching, and we'll also discuss antibody diversity and its generation. Now we've been talking about the structure of immunoglobulin genes, and we did talk about the specificity of antibodies. And now the question is that we know for sure that there is something called non-immunoglobulin genes. So we need to have basic understanding of how genes are expressed and what is the basis of diversity that we normally get in terms of expressions of different amino acids on top of cells as receptors and Fab as a part of immunoglobulins. Now remember, the human genome, that is the DNA that we inherit, has been now sequenced and probably considered to have somewhere about 20 to 25,000 genes. And within one mammalian species like ourselves, we contain similar number of genes. Now the question is that there are only few genes that produce so many different antigen receptors that we normally see. And what happened is that the strategy that the body has is that it involves combination of genes. So they are combination of different genes to achieve the required degree of diversity. So we need to understand three important aspects of the gene. Number one is that there are variable and constant region of AIG, immunoglobulin gene, and they are all coded by different gene. Number one, there are different variables we call V regions, and they can be linked up to a single constant region. And then again, there could be something we call antibody genes which are rearranged. So we have a combination of different genes that can be put together. And this process of rearrangement during differentiation will bring a set of genes that we code as V and C regions. So what you need to know before we go further is that the genome of an individual contains linear arrays of gene. For example, if you pay attention to this one, you will see a DNA strand, and then this DNA strand is transcribed into RNA, and then RNA is translated into protein. So that's pretty much the sequence of event in a typical case. And some basic information that you need is that every diploid cell in a human body that contains similar number of genes, and the only exception is lymphocytes. And what we're trying to say is that cells within an individual, they differ from one another because they transcribe and translate different sets of genes. So we talk of expression of genes. So one is a presence of gene and one is an expression of gene. Now we also have to keep in mind that there is a gene expression and then there is a control of gene expression. So the activation of different cells, they basically are controlled by gene expression at multiple levels. So there could be many transcription factors which basically are protein that would either begin or modulate, and they basically are generally bound to regulatory DNAs, and they are closer to the 5 end of the ladder, as you can see over here. And then again, there is a rate of transcription, there is a half-life of messenger RNA. All these basic mechanisms of genetics have to be understood so that we can understand how genes are regulated. Now also remember, just in this figure is that, as you can see, exons, most genes that are coded for a protein have a characteristic structure of exons and introns. And exons are basically sequences of base pairs that are later transcribed into mature messenger RNA. And exons are, in this case, you can see, are separated from each other by those two lines appearing. They are introns. Now, you know the basis of genetics, that when a gene is transcribed into RNA, the entire stretch of DNA, exon plus introns, are transcribed into a primary RNA transcript. So you can see from here, there's a 3 end, there's a 5 end, and there's a transcription of exon 1, 2, 3, and 4. So they are being transcribed into primary transcript RNA. Now, once it is done, then we have specific enzymes that would splice them. So they would splice the introns which are non-coding. So they're non because they don't express any code. So they kind of cut them out and bring them together. So what you will see is that the introns disappear, and then you have a mature messenger RNA, and then obviously mature messenger RNA in terms of exon 1, exon 2, exon 3, and exon 4 are then translated into a protein, and you can see a full protein over here. In this case, it's very obvious. And then you also would see in the genes, when they are coded for protein, they are expressed at the cell surface, and they have a leader sequence on the left of the mature messenger RNA, which is at the 5 end. And then this would code for the sequence of about, I would say, 10 mainly hydrophobic amino acid called the signal peptide as the amino terminal of the protein. So there's a carboxy terminal, COOH, and there is an amino terminal in this case. Now, if you look at the genetic events in the synthesis of immunoglobulin chain, so you will see firstly organization and rearrangement of light genes that we will discuss in figure 6.2 and 3. And then again, we'll talk about the organization of and rearrangement of heavy chains, and then we'll talk about how these genes are regulated. Now, let's pay attention to gene rearrangement and subsequent steps in the synthesis of kappa light chain. So we have a kappa light chain in this case, and you can see from here that there is a germline DNA, and germline DNA has 3 important portions. You see a 3 end on one side, a 5 end on the other side, and has 3 important portions. There's a V portion, variable, there's a J portion, which is joining, and then there is a constant chain, which in this case happens to be kappa. And now, as we discussed earlier, that when the germline DNA is transcribed, it is rearranged. So you still have 3 end and 5 end, and we see that out of different combinations that we have in variable portion of the sequence, we pick up V1, V2, and then in the J sequences, where it's like 5 different sequences that we have, as a matter of fact, in V we have roughly about 40 different sequences. In J we have about 5, so we pick up in J only 2, J4 and J5, and of course C will come as such because that will be the heavy chain. And then this is transcribed into this case into a primary RNA transcript. And then again, you will see that we are losing introns and we're putting them together. V2 joins with J4 and then J5. And then again, as we discussed a while ago, they are specific enzyme. They will splice it up and bring these together, and then we will have a mature messenger RNA, and obviously that will be translated into a two light chain. So you can see from here terminal, amino terminal, and we'd see a carboxy terminal. Now let's move to the involvement of Vdjrecombinase in rearrangement of DNA coding for kappa light chain. So remember, this is a specific enzyme that we have, Vdjrecombinase, and this is a very complex enzyme, and basically it mediates the rearrangement of receptor genes in B and T cells. And as the name suggests, this is a product of two recombination activating gene. It is called RAG. R stands for recombination, A for activating, and gene, of course, is gene RAG, RAG1 and RAG2. And they are very critical in initiating the recombination in lymphocyte precursor cells. So we have both RAG1 and RAG2 proteins that basically are required in the first stage of cutting a immunoglobulin and then of course a T cell receptor DNA. And as we will see in clinical studies, that sometimes these genes are not present, or we have an animal model which is called RAG knockout mice, where the mice would not have expression of these genes. And if you don't have these genes, so obviously you will have a deficiency of both B and T cell. Now also remember that this specific recombinase complex enzyme, it also includes genes that are involved in the repair of DNA strands. And especially when the lymphocytes are developed, so we'll talk about the precursor lymphocytes, and when they get developed, there's a sequence of events. That's where we have to have RAG1 and RAG2. Now, this is another figure over here that you will see that has a gene rearrangement and subsequent steps in the synthesis of human heavy chains. And you will again see pretty much the same sequence. The only difference is that now we're talking about a heavy chain. It could be a micro or it could be epsilon. And again, if you pay attention to the germline DNA, which is rearranged to begin with, so we have a 3 end, we have a 5 end, we have VDJ segments. In V segment, we have a choice of about like 50. In D segment diversity, we have about 9 choice of 20. And in the J segment, we have 6. And then again, these are the C region genes where we'll pick up either gamma, alpha, micro, delta, or epsilon. And then again, when it gets resorted, rearranged, in this case, you can see again, V2 gets picked up from V section, D2 gets picked up from D section, and then J5 and J6. And then we have pretty much everything in terms of C region. But still, it is a beginning of a B cell DNA. So cells are moving towards a B cell. Also remember, the B cells are the one that will make antibodies. Now, this B cell DNA is then transcribed into primary RNA transcript. So we see that we have V2, D2, J5, J6, and then we pick up micro or epsilon. So we have at least two over here, and the rest of them are not picked up. So that will form the primary RNA transcript. And as we move up, then we have the next step, which is called splicing. In this case, we have alternative splicing. So you can see that V2, D2, and J5 in both cases is split. One attaches to micro and the other attaches to epsilon. And once they are translated into protein, we end up a micro or epsilon heavy chain polypeptide. So we have VH, and in this case, we have a C for micro. Then again, we have VH, and then we have a constant segment for epsilon. And then, of course, we have an amino end, and we have a carboxy end. The other important thing that we will discuss is that, as we talked about that some of the antibodies have the ability to class switch. So there's a class or isotype switching, and we need to find out what is the mechanism of class switching. So S, basically, in the new figure that I'm going to talk about, S stands for switch region. And you can also see that in this case, we are talking about upstream of each heavy chain constant region. Now, if you pay attention over here, we have a 3 end, we have a 5 end, and then again, we have a V segment, J, and then we have a constant region gene. So there are a bunch of constant region genes, and within those genes, as you can see, that we have switch regions. So whenever the message comes to B cell that we need to switch from one particular antibody production to another antibody production, and of course that message will come by under the influence of T cells. So we will have a B cell DNA after the class switch. So you can see that switch from one S goes to another S. So we will have again V2, D2, J2, J5, and a joining segment, and then we will have that jump of switch region, and we'll end up with picking up one of the alpha 2 in this case. And then of course it can be transcribed, and then in transcription 3 end and 5 end, further splicing that will get rid of all the introns, and then we will end up with messenger RNA for, in this case, you can see that we end up with messenger RNA for gamma 1. So gamma 1 is a messenger RNA in this case because we are looking at the structure of, so that kind of a switch that we initially were going for alpha, and then we ended up with a gamma 1 because of the switch, is the beginning of the concept of class switching. Now, if you pay attention to how the receptors are edited in B cell lineage. So once we have a B cell lineage, so we need to understand that it can go a second rearrangement of its light chain. So after it has formed a recombined VGH unit, it will begin something we call receptor editing. So the receptor at the distant end has to be edited. Now in this figure, if you look at somatic gene conversion that generates diversity in immunoglobulin genes of survival species, you will see the figure illustrate the phenomena in basically chicken hemoglobin heavy chain locus. There's a short sequence of DNA from one or more pseudogene, and they are copied to that. So if you pay attention to about like 20 pseudogenes, and then there is a functional gene segment, again pretty much the same as we have already been used to. There is a VDJ segment, and there's a constant segment, and then it is linked with those pseudogenes, and this is called a germ DNA. And when we have VDJ recombinase activity, so we end up joining VDJ together, and that will basically form the transcript for B cell DNA. And in this case, what you can see is that we have this ability of going into that VDJ segment and edit it. So this gives you a repertoire where we have a huge library of B cell lymphocyte, and we can pick up all different specific epitopes, and then we'll have a diverse epitope, and that will give us a ability to make different types of antibodies. So we can estimate that if we have about thousands or multiple specificities of immunoglobulin, within one individual we can generate in order of about 10 to 15, and then again we can increase that to somatic hypermutations. So you can see that we have this enormous capability of synthesizing a different type of immunoglobulin molecule, and each of them will act as a B cell receptor. And to summarize that, so we have the heavy chain, which has a portion which is called VDJ, and V stands for variable, D stands for diversity, J stands for joining, and the V portion then again has VL and VJ. So in the DNA from every cell in the body, it contains multiple VDJ genes, and we can sort it out and make a different set of arrangements. So different, and that during differentiation, B cell rearranges itself into a heavy chain DNA to join one variable and other, diverse H part. Now, this receptor editing basically is important because this is a mechanism where we can rearrange DNA of a very particular B cell, and then we can come up with unrelated elements from V1 and J5, and then which can be used later in a second rearrangement. So receptor editing can occur when a B cell with a specific receptor specific for a self antigen interacts with a self antigen. Well, one outcome of this interaction is that V and J genes of B cell undergo a second rearrangement, which may generate a Vdjrecombination unit specific for a foreign rather than a self antigen. So in a nutshell, what we are trying to say is receptor editing may increase the diversity of the overall response to foreign antigens. So that's why we need that. Now, summary slide for class switch recombination. We know once antigen stimulates the production of antibody, there is a rearrangement of heavy chain DNA, and then we have VDJ unit that join together to form either micro or delta, or they can basically rearrange, especially for the C region. So they can either go for gamma, go for alpha, go for epsilon. So that's what's happening over there. And also remember that B cell has this capability to synthesize IgM and IgD, which initially had, because IgM and IgD, if you remember from that mnemonic slide, so they are like smaller antibodies. They want to hang on to their mom B. So they basically are producing M and D. And now, because of the class switching recombination, they can also have this ability to produce IgG, IgA, or IgE. And the good thing is that the antigen which stimulated the production of IgM and IgD, now the same antigen specificity now is going to produce IgG, IgA, or IgE. So it's very antigen specific. Now, the idea basically in this whole concept of genetics of genetic basis of antibody structure is that we have inherited genes for variable regions for both light and heavy chains, number one. Number two is that we have rearrangement of VDJ segments, and different combinations and random assortment of heavy and light chains can be done. Number three, we have this ability, a junctional diversity at the sites of VDJ. So if, for example, antigen stimulates and we have somatic hypermutation, so this would basically lead to even increase in the diversity of the overall response that we may get to a foreign antigen. And then also remember that this somatic gene conversion in species other than human or mice. Now, these different mechanisms that we have discussed will allow us that we can have number of genes and generate a vast number of antibody, each one of them having a different type of receptor or antigen specificity. Now, when we say we can generate a diverse group of antibodies, what we are saying in immunological terms is that there is a presence of multiple V genes in germline. We have VJ and VDJ combination associations available. There is a random assortment of heavy and light chains. So you can see that all these tools that we have of junctional diversity, of somatic hypermutation, of somatic gene conversion will allow us that we basically can create different sets of antibody. And recently, in the last probably couple of years, we have also identified an enzyme, and it is called activation induced cytidine deaminase, AID. And that actually plays a very important key role in initiating class switch recombination. So that is very, very important, and actually has three important roles that this enzyme initiates, because that's crucial. Number one, it causes class switch recombination. Number two, it causes somatic hypermutation. And number three, it causes gene conversion. Now, as the name implies, AID removes cytidine groups from the DNA in activated B cell to form uridine. So this will generate a UG base pair in DNA. The mismatched base pairs are then removed by one of the several different enzymes. And this will result in changes in DNA, especially in the V region of immunoglobulin. And that's why AID wants to interact specifically or preferentially with the immunoglobulin V region. And these genes pretty much are currently very well understood. So remember, there is a definitive role of AID, activation-induced cytidine deaminase, in the class switching. And then again, as we go along, we will find out what are the other things which are available for us to control or allow the body to class switch. So I will just summarize some of the key concepts and then we'll be done. Remember that we have a capability of synthesizing enormous number of different immunoglobulin molecules. That's number one. And each of them can attach to a B cell and is specific for a particular epitope. Number two, as I've been saying over and over again, the variable region of H gene in an antibody molecule or Ig molecule is coated by three separate genes. And we have referred them as V variable, D diversity, and J as gene segments. Now, a distinct gene segment codes for a constant region of a heavy chain, CH. And the variable region of the L chain is coded by two genes, and we talked about that. And DNA from every cell in the body, the germline, contains multiple V, D, and J segments for us to synthesize heavy and light chains. Now, when B cell goes through differentiation, a B cell rearranges its heavy chain DNA to join one of the V gene segment to one of the D gene segment and one of the J gene segment. So this combination of V, D, J unit codes for the entire variable region of the heavy chain. So these genes are rearranged, put in the VDJ unit, which are close to heavy chain constant region of basic, micro, and epsilon. And then again, we have a rearrangement in the same type, which produces a gene unit coding for entire V region of IgL chain. So the important thing that we need to understand is that why do we have diversity in antibody production? Why is it that diversity in antibody specificity, how is it achieved? How do we get that? So there are a couple of things that we have discussed in the whole lecture, and I think if you can remember that and you can go over this lecture again and read the book, you will realize that diversity in antibody is achieved by many different mechanisms. Number one, and it's going to be repeat for you as well, there are multiple inherited genes for the V regions for both light and heavy chains. Number two, rearrangement of V, D, and J segment in different combination and random assortment of heavy and L chains. Number three, how do we get junctional diversity? So we get junctional diversity at those specific sites for V, D, and J gene by joining that is caused by, we call it imprecise joining. So there is either an imprecise joining, there are deletions, and then there are TDT mediated nucleotide insertions. So these are some of the events that would add on to junctional diversity. And then we also know that whenever there is a gene hypermutation, we call somatic hypermutation, and this normally occurs after stimulation of antigen. So we have an antigen entry, and then there's a somatic hypermutation that will lead to selection for mutation that basically will allow the antibody with higher affinity for the antigen. So that's another thing, that what the body is trying to do is it will pick up an antibody that will have more attachment to a particular antigen for an immune response. And finally, we did talk about somatic gene conversion that happen in species, but not in human and mouse. So we have short DNA sequences from non-rearranging genes, and they are copied into rearranged VDJ gene unit. And in the last slide, as you saw, that we talked about these mechanisms, but I wanted to lay emphasis on one enzyme. Of course, there are more than one, and we are going to talk about that as we move along in this initial series of basic immunology. One enzyme I said was activation-induced cytidine deaminase, AID, and that plays a key role in initiating class switch recombination and somatic hypermutation and gene conversion, which basically are three major, these are the three major pathways that allow us to generate tons of antibody. And as I said, something to the order of 10 to the 15. So we can have all the antigens in the world, and then we can also make very specific antibodies. And these are, of course, three important mechanisms that we have. I repeat, somatic hypermutation, and I said earlier that always happen after the antigen exposure. And then we have gene conversion, and then we have classic switch recombination. So you pretty much, depending upon whichever order you want to memorize. So these are the three important things that stand out, especially for this lecture. And I would also want to emphasize in the last part of the lecture that the importance of RAG, because this is some term that I want you to remember. This is basically product of two recombination activating genes, RAG1, RAG2, and they are very critical in initiating recombination in lymphocyte precursor cells. And you will wonder, why do we care about the precursor cells? Well, we do care for precursor cells. That right now we're talking about basic immunology of the production of B and T cell and the genes that regulate them. But also remember that once we go in tumor immunology, there may be B cell tumor, there will be T cell tumors, and then we need to stop that. So that's another important thing that we have to keep in mind. And this specific concept, whereby when it was found that the antibody genes can rearrange themselves within the genome of a differentiating cell, that basically got Tonigawa, the Japanese scientist, to win a Nobel Prize in 1987. So you really have to understand the concept behind that. And if we do understand the concept behind that, it will be very easy for us to understand why do we have and why do we need antibody diversity and what is happening in this case. And one last thing I thought, maybe if you have a couple of minutes left, that want to emphasize is just for a concept. When we say presence of genes, we talk of expression of genes. So remember, the expression of a specific pattern of gene basically would determine the cell's function. So we are worried about the cell function. So each one of our cells have a function to perform, and basically it is coming from the genes. Now, your book gives an example that, for example, every cell contains an insulin gene. So only pancreatic beta cells express that gene. So these cells which express the gene have the capability to make insulin. Similarly, all the cells, they have immunoglobulin gene. All of our cells have immunoglobulin gene. But by the end of the day, only B cells, and of course they're differentiated from plasma cells, as I discussed yesterday, they are the ones that express immunoglobulin genes, and therefore they have this ability to synthesize immunoglobulin molecule. Like all of the cell, except B cells and T cell, contain immunoglobulin genes, but do not express that. So you got an idea? So all cells have DNA, all cells have this sequence genes over there, but they do not perform that specific function because these are not expressed. They are there, but they basically are not expressed. And once they've been expressed, and then they can go. And that also bring into picture that the control of gene expression. So if gene expression is going to determine the function of the cell, how do we control that gene expression? Because we don't want all the cells in the body to start producing insulin, or we don't want the pancreatic beta cells to stop making insulin. So these basically activity is controlled by transcription factors. So these are transcription factors, which are protein that initiate or modulate transcription, generally by binding to regulatory DNA sequences close to 5 end of the genes that we discussed earlier. So the rate of transcription and the half-life of messenger RNA are also important. So the whole idea is that if we understand the mechanisms that regulate gene expression, and especially we want to find out how these genes are either turned on or turned off in different cell types, this is a very important area, and it's a topic of very intense research, and that is happening these days. So I hope that I've tried myself to give you the idea that the genetic basis of antibody structure is important, and we have discussed that there is an enormous diversity of the immune response. We have focused on diversity of antibodies, and we have also focused on diversity of immunoglobulin. We talked about immunoglobulin superfamily as well, and I told you that also have MSC, that also have T cell receptor. And also remember that immunoglobulin molecules that are secreted forms of antigen-specific receptor found on individual B cells. So the immunoglobulin molecule, as we say antibodies, basically are secreted and then they are expressed on top of B cells and T cell with sometime with different antigenic specificities. And also remember, as I said earlier, that estimates of the number on B and T cell with different antigenic specificities that can be generated in a single individual range from somewhere to 10 to the 15 to 10 to the 18. So if I was to use some other words or your book's word, all of us have the ability to generate this number of different immunoglobulins or T cell receptor molecules. So you can imagine that how profound and robust response do we have in our system, where we have this ability to generate very specific responses. That's why we want to understand that. And as I said yesterday as well, that it is one of the important objective and goal for us to understand that. And I would encourage you to please go and read this chapter from beginning to end. You can come and ask me any questions. And then again, once we go for development of B cells and development of T cells and the biology of B cell and T cell response, then you will figure out why is it that we are interested in this class switch. Why is it that we are interested in knowing that there has to be very specific and diverse response in terms of antibody production and in terms of immune response. And remember, as I said the other day, that so much progress has been made in immunology that by the time we teach you something, things are already obsolete. They're old, and they are coming with newer ways of understanding, new drugs, and so on and so forth. So I encourage you, I encourage myself as well, those of you who are really interested, and those of you who want to know how an immune response is initiated, and those of you who are looking into like practice even pharmacy 10, 20 years from now, as one of our professors used to say, that you need to teach your students that they need to practice pharmacy in 2025. So pretty much, we really have to stand up to this challenge and get to the basis of immune response. And thank you for your attention and have a good day.